We have two talks today, and we're really delighted to have Dr. Gu Feng and Dr. Stephanie Moquin from Novartis Institute of Tropical Diseases, and they're calling in from the West Coast of the US, and as well as Dr. Panisadi Avirotnan. And they're going to describe two their works, their respective studies that they have been working on developing antiviral drugs for dengue. Both these studies were published recently in very high impact journals, so we're delighted to have the authors to give us their overview of their findings. Without taking too much time, I'm going to hand this time over to Dr. Gu Feng and Dr. Stephanie Moquin. Please take it away.
Thank you so much, Eong. It's great to see you again. Eong is also a great friend and a long-term collaborator. And yes, I would really like to thank the meeting organizer to invite us for this very exciting event and share our story with the rest of the group. And yes, I'm so happy to talk about our work on NITD688, a new antiviral against dengue. As Eong mentioned, that this is a work from NITD. NITD stands for Novartis Institute for Tropical Diseases. NITD is located currently in the San Francisco Bay Area, but it was actually founded in Singapore and has very long history in Singapore. Until 3-4 years ago, we moved to the Bay Area. And this molecule, NITD688, was really designed, developed in Singapore. So I'm really honored to talk about the story for you today. So next slide, please. So I just want to reiterate how important dengue is. It is really an increasing threat. And the dengue outbreak, especially throughout the COVID pandemic, made it very clear that it's a further burden on the already overwhelmed healthcare system. As you can see that WHO has reported over more than 10 times higher increase of outbreak in the last 2 decades. So this is really a problem that we all need to tackle. So next slide, please. I think the unmet medical need is very clear. It's endemic in 100 countries, 100 million symptomatic infections, and even the death has peaked in 2019 to the 36,000 deaths. And it overall represented the economic burden of about $8.9 million US. And there is no disease-specific antiviral therapy, but there is advancement in vaccine research. As you know, that Sanofi vaccine is Demaxia is accepted in some countries, even though, and also the Takeda vaccine is providing better protection, as the group is seeking registration for TAC03. However, they are not covering all 100% of the population, and it yet doesn't have 100% of efficacy. And also there is reported Wolbachia infected mosquito and transgenic mosquito providing very good protection in the outbreak area. But we believe that there is a need also for antiviral so that we have multiple pronged approach to overcome this disease. Next slide, please. So our approach in dengue is actually derived from our understanding of the disease pathology. As you can see that on the right figure, it was a report from Daniele Librati early in 2002, and the viral load in the dengue fever and the viral load in the dengue hemorrhagic fevers are different. There is an early 1 to 1.5 log difference in the viremia in the early onset of fever. So we believe that reducing the viral load peak early in the onset of the fever can really accelerate viral clearance in order to reduce the disease symptoms and disease severity and transmission. So that is our guideline as basis for antiviral drug development, drug research principle to get to a specific antiviral so that we can kill the virus early and reduce the viremia early. So that's our principle. And next slide, please. So this is the history how we came up with this compound. We have carried out high throughput screening with about 1.5 million compounds. And this has been done in the dengue 2 infection in the HUH7 cells using a CPE assay. The CPE assay is cytopathic effect assay. And through these high throughput screening and reconfirmation, we selected 160 hits, which actually are the compound which could inhibit dengue virus in this assay. With 160 hits, what we did then is to run these hits on dengue 1, 2, 3, 4 assay and also chikungunya assay using a different orthogonal assay, and that is the HH7 high content imaging assay. And this is to detect the viral antigen in these cells. And through these 160 hits reconfirmation, we want all 4 serotype N active compound and compound that is not active in chikungunya, so that we can eliminate some host factor and that we can select very specific antiviral inhibitor. So through this filter, we end up with 3 classes of hits that are specific against dengue 1, 2, 3, 4. And then on these 3 classes of hits, we carried out extensive medicinal chemistry lead optimization to make it very orally available to dose by oral route and to have the safety that is satisfactory for dosing in human. So with this, we end up with 1 compound, and that is NITD688. Next slide, please. So here is the compound on the left, on the right-hand side, you can see the structure of this compound. This is all published, and this is a novel anti-dengue antiviral, which has all 4 serotype activity. And the nanomolar, I'm just giving you a summary of this, but I will go through all these data in the detail. So the serotypic activity ranges from 8 nanomolar to 38 nanomolar across all 4 serotypes, and it is efficacious in the mouse viremia model in acute and in delayed treatment mode. And through the resistance study, which Stephanie will talk to you in a second, we found that the mutation actually came through in the transmembrane 5 domain of NS4B, depicted on the bottom of that slide. And so we could raise resistance and find interaction of this compound directly with NS4B. So we believe that this compound inhibits viral replication through its interaction directly with NS4B. And so we also carried out safety studies, and we found some liver toxicity at high dose in both rat and the dog. We have sufficient safety margin, and the finding in the liver, which I'll explain to you later, is mild, moderate, and monitorable and reversible. So this compound has been through IND enabling studies, and the IND has been filed with US FDA, and we got the safe to proceed just last Friday. So our phase 1 will start in June in the US, and phase 2 in the dengue fever patient, we believe, will be happening soon in 2022 in Asia and or maybe Latin America. So next slide, please. So basically here, I'm going to present you the full profile of this compound, NITD688. As you can see in the left table, we list dengue 1, 2, 3, 4, EC50 using the high content imaging method in A549 cells, and that is from 8 nanomolar and in dengue 2 and 3 to 38 nanomolar in dengue 1. And we also use different cell type, HH7, and using different detection methods, such as plaque assay and CPE, to confirm this in both primary blood mononuclear cell, which is the primary target of the human cell, to confirm the activity is real. We also tried in chikungunya cell that is not active, as you can see, or it's very weakly active. And the cytotoxicity is not, it's very non-cytotoxic based on CC50. We also tested in the relative other flavivirus, such as Japanese encephalitis and West Nile yellow fever Zika virus, and the Pengwasan and Usutu virus. And as you can see on the right side of the table, all of these virus is not specific. There is no activity. And you will see the reason for that in Stephanie's talk in a few slides later. Basically, our conclusion is that NITD688 is just specifically efficacious against dengue virus. It's not active against any other virus in the flavivirus family, and it's not active against alpha virus. So now I'm going to hand over to Stephanie to talk about the resistance and the interaction of NITD688 with NS4B. So I'm going to hand over to you, Stephanie.
Thanks, Gu Feng. Can you hear me?
Yes.
Excellent. Can we get the next slide, please?
Yes, we cannot see you.
Oh.
Can you start the video? Sorry.
Yes, no worries. Sorry, we're switching here in the middle of the talk. And can you see me now?
Yes, that's perfect. Thank you. And I've switched slide.
Got it. Excellent. Yes, so because we used a phenotypic screen to discover these compounds, we actually didn't know the target when we started, as Gu Feng said. And so in order to figure out what the target was, we passaged virus with increasing concentration of compound. It was actually a precursor to NITD688, but it was in the same class. And as you can see, after 13 passages, we did an EC50 test, and we saw that the EC50 shifted greater than 10-fold. We sequenced those viruses, and we found the mutations appeared in the viral protein NS4B in 3 different residues, T195, T215, and A222. As you can see, A222 was selected for twice, as well as T215. Next slide, please. When we looked at the conservation of these 3 residues in the NS4B protein, well, first of all, we saw that they were in the transmembrane domain in the loop between TM4 and TM5, as well as in TM5. And when we looked at the conservation, we saw that it was very strongly conserved across the dengue viruses. You can see dengue 1, 2, 3, 4, it's strongly conserved. But it's not conserved across the other flaviviruses, which may be why we have activity of NITD688 across all the dengue viruses, but not across the other flaviviruses. Next slide, please. Next, we wanted to know whether these mutations caused any deficiency in replication and whether they conferred resistance to NITD688. So for these experiments, we used a dengue replicon where the structural genes are replaced with a luciferase gene. And we transfected the wild type replicon as well as the NS4B mutant replicons into A549. And we looked at their replication over time using luciferase. So on the left-hand side, you can see that wild type is in black, and all the mutants display a little bit of decreased replication efficiency, somewhere between 36 to 77%, I believe. So they're a little bit deficient in replication. And on the right-hand side, you can see that when we treat these replicons with NITD688, we see a large shift in EC50, somewhere for the single mutants, somewhere between approximately 1 to 300. And we see a much greater shift, about 1,500 for the double mutant, suggesting that these compounds, that NITD688 does indeed target NS4B, and these resistance mutations do confer resistance to the compound. Next slide, please. And lastly, we did some biochemical experiments. We purified both wild type NS4B as well as double mutant NS4B. We chose the 2 mutants that appeared most frequently. So we made these 2 proteins, we expressed them and purified them in E. coli. And then we incubated these proteins either with a controlled DMSO or with NITD688, and we examined these spectra using NMR. As you can see on the left-hand side, we have wild-type NS4B in black, and in red is the spectrum when you incubate it with NITD688. And in the little zoom-in box, you can see a specific peak is broadening and shifting, demonstrating that NITD688 is directly binding to wild-type NS4B. And if you look on the right-hand side, you can see the spectrum for double mutant NS4B, again in black, and then in red when it's incubated with NITD688. And we do not see any peaks shifting, demonstrating that NITD688 does not bind the double mutant, suggesting this is likely the mechanism of resistance. And with that, I will pass it back to Gu Feng to finish up the story.
Thank you, Stephanie. Yes, next slide, please. So basically, I think we understand that to some extent that this compound is through the phenotypic screen, through the resistance study, has very good in vitro potency and interact with NS4B. And we just also now want to make sure that it works in the animal model. And now dengue virus is highly evolved to infect human. It actually doesn't infect other animals very well. And also because there isn't yet a approved successful antiviral drug developed so far in the clinic, there is no really a true animal model that we can translate preclinically to fully validate that model. So basically there are several available models as I put on the slide. There is the mouse model and there's the non-human primate model. And none of these is fully validated, but in the mouse model, wild type model has high injection, which shows some high level of viral load, and which shows some symptoms and death. And there is also the AG129 mouse model, the interferon alpha, beta, and the gamma knockout mouse model, which allow actually both the TSV01 strain of virus or D2S10, which is a mutant strain virus, and to get to very high infection and cause some death. And so we chose to use the AG129 interferon alpha, beta, gamma knockout mice, and just to have this viremia model, because we believe that our compound is antiviral, and as it's antiviral, we want to have a viral readout to prove this mechanism. And so that's what we did in our next experiment. So next slide, please. I just want to show you how the experiment was organized. We have these two treatment models. The top one is the acute treatment model where the virus is infected at the same time as we dose the compound. We call this acute treatment. And then we wait for 3 days, and every day we'll dose 2 times BID. And then at the end of the day 3, and we will sacrifice the mouse and measure the viremia. And the second model, the mode of treatment on the bottom is this delayed treatment. So we would infect the mouse, but we will not treat the compound. We will wait for 2 days and start to dose the compound only 48 hours post-infection. And then we will take also everyday treatment, everyday mouse blood and measure the viremia, and every day, and until day 5, we will sacrifice the mouse. So this is 2 different modes of treatment, one acute, one delayed treatment. We want to look at NITD688 efficacy in this viral load reduction. So next slide, please. So on the left panel, you can see the 3-day acute treatment mode where NITD688 was given exactly at the time of infection in the mouse model. And the black dots is different, it's all the control vehicle control, and the different colored ones is at different dose given and at the with NITD688 treatment. As you can see, when we go higher and higher in dose, up to 100 milligram BID, this green group, you can reach really up to even higher than 3 log of reduction. So there is a clear dose-dependent reduction of viral load when we go higher and higher in dose with NITD688. So this is very encouraging. But we know that in the infection setting, the patient would never come in at the onset of viral infection. So we try this 5-day delay treatment, as I explained earlier, where NITD688 is given 2 days post-infection. So there we use one dose, which is 30 milligram per kilogram, two times daily. And we look at day 3, day 4, or day 5 post-dosing, and how much it's different in the treatment group versus the vehicle control. So on the day 3, there is some reduction, but not as significant. But starting from day 4 and day 5, we can see a very clear significant difference between the treated and untreated group that is more than 1 log. So with this treatment regimen, we feel that it's going to be relevant for human dosing for the dengue patient who can come in early onset of fever, and which can be treated using NITD688 to reach a significant reduction using NITD688 compared to natural untreated patient. So yes, next slide, please. So with that efficacy, we then continue to look for safety of this compound. Of course, we ran a lot of in vitro safety in different essential channels and enzyme and receptors. There's no flag. We also saw that it's clean in genotoxicity, phototoxicity, and but most importantly, we tried them in vivo toxicity study that is in rat and a dog. NITD688 was well tolerated in 14-day repeat dosing oral toxicity studies, and leading to a NOAEL establishment in both rat and dog. So NOAEL stands for no observed adverse effect level. So there is a clear safety level for these 2 species with NITD688. And at higher dose, we actually do observe there's toxicity in liver. And what we see is portal infiltration and bile duct hyperplasia. This is associated with liver enzyme and bilirubin elevation. As I mentioned at the beginning, these findings were actually quite mild to moderate at very high dose, up to 500 milligram per kilogram. And they are also monitorable because of the liver enzyme change, and they are reversible when we wash out and withdraw the drug. So based on the safety profile, we feel that there is sufficient safety profile of this compound for us to support this compound moving into the clinic. And the human efficacious dose are far below rat and dog, no observed adverse effect level. So next slide, please. Yeah, I also want to show you that NITD688 has very good oral PK profile. And so the figure A and B is in red. You can see the oral exposure, and blue in the intravenous dosing exposure. As the 2 lines are very close to each other, that actually shows us that it has good absorption by oral route. And based on this compound's PK profile, it has low clearance, long half-life, and good absorption by oral route. And it's also consistent in rat and dog. We have carried out more species PK and used that different species, we can allometrically scale to human PK. And on the bottom C and D figures, you can see a modeling and simulation of how this compound would behave in human by oral dosing. And by 6-day oral dosing, you can see consistent coverage of exposure of this compound. And we also try to project a human efficacious dose by targeting at least 1 to 2 logs of reduction. So we want to have the C trough, so the minimum level of the NITD688 in the blood of the human, never below 3 times or 10x EC50. And so that gives us about 35 and 150 milligram dosing, which can maintain this level, never dropping below 1 to 2 logs of reduction in virus. So that is our current efficacious dose range that we think we will achieve the significant viral reduction. So next slide, please. So to summarize all of this work, it's being published, all these data that Stephanie and I showed you in the Science Translational Medicine early this year in February. And if you just Google NITD688, you will find this paper with all the detailed experimental detail and result. So feel free to look for the paper, and if you have any question, feel free to ask us. So next slide, please. So at the end, I just want to also bring to your attention what we are thinking of the use case of this antiviral drug in the future. As we think about all the people, all the patients who have dengue in the field, we designed different target product profile. And it can be prophylaxis dose, can be chemo prevention, it can be acute treatment of dengue, it can be treatment of severe dengue. But we look at all these different target product profile, and we consulted some key opinion leaders in the field. We view that the early treatment of mild to moderate dengue could be our top priority, and it could be our top priority target product profile, which has the most impact of an antiviral. And this is also thinking of how COVID is affected so many people and how we could use a anti-COVID drug early in the infection of majority of the people, which would be able to reduce their symptom and reduce their progression to the severe disease and prevent death. That is pretty much what we think is going to be the most useful use case of an antiviral drug. However, this does come to a hurdle that we feel that is worth discussing with all the experts here as well, that we do need to get the dengue patient quite early and in their onset fever, as there is only a short window of the high viremia in day 1, day 2, or maybe day 3 after their onset of fever. So with that, maybe a rapid diagnostic kit, which is robust, low cost, accessible for all endemic area, will benefit the deployment of the antiviral, and whether we are making already sufficient effort in the rapid diagnostic kit is something which we can further discuss. But we do believe that if there is an antiviral paired with a good rapid diagnostic kit, it will maybe encourage patients to seek treatment early and will maximize the impact of the antiviral, such as NITD688, to majority of the dengue patients. So with this, I just want to go to the last slide, which is the acknowledgement slide. And so Novartis is definitely the main sponsor of this study. And a lot of the work has been done back in Singapore when NITD was still in Singapore. And a lot of the team is still remaining in Singapore. And for that piece of work, we are very grateful for the support of the Singapore Economic Board, Development Board. And after we moved to California, we would like really thank Wellcome Trust and NIH in the US to continue to fund our project, and also the collaboration with Utah State University for all the other flavivirus antiviral testing, and also WuXi AppTec, who did a lot of CRO chemistry help in our early stage of NITD drug development. Thank you so much, everyone, and welcome any question.
Thanks very much, Gu Feng and Stephanie. There is one question. I was going to hold everything till after the second talk, but there's one question that's directly related to your presentation. Maybe we'll just take that and then we'll move on from there. Question is, what methodology was used to filter the 160 hits from 1.5 million compounds?
Yes, I can answer that. I pulled up our flowchart. So the first screen, the 1.5 million screen, was a single point screen. So one point for each compound. And we chose our top 1% from that and did a dose response. And that yielded 13,000 compounds into a dose response, and we selected anything with an EC50 less than 5 micromolar, and that basically yielded the 160 compounds that went into the all 4 dengue virus HCI assay.